Hello, this marine ecology lecture is about secondary production. It follows the one about primary production. Secondary production is related to primary production, but it's a little bit different. So we'll start out by defining what secondary production is. Then we'll talk about how we measure secondary production. I'll review 3 of the common methods used to measure secondary production. Then we'll talk about some of the factors that control the amount and rate of secondary production. And we'll begin to relate this to the food chain and the concepts of trophic transfer and biomass pyramids. So primary production is the production um, of organic matter using inorganic ingredients. And so to do primary production, you need to be a primary producer, something like the grass in your lawn or the phytoplankton cells in the ocean. And these organisms take raw ingredients, inorganic ingredients from the environment like carbon dioxide, water, and chemical nutrients, and they use some energy source, sun in the case of photosynthetic organisms, and they turn those raw ingredients into organic matter, which you can see visually in the growth of your lawn, for example. This symbol, number symbol 1 with a degree symbol behind it, that's a shorthand for primary. So if you see that in the notes, that should mean, pronounce that to yourself, primary. So if primary production is the production of this plant life at the bottom of the food chain by the primary producers, um, secondary production, then you might infer is the production of animal life. So what's an animal that might consume the organic matter produced by these primary producers and accumulate organic matter in its own body? How about a sheep? So sheep are one of a number of terrestrial animals that eat grass. And so basically the production of the sheep, its meat, its wool, its milk, and all the things that a sheep makes, that would be considered secondary production. And you could use the 2 degree symbol to indicate secondary production. So you have to have primary production first to have secondary production. Obviously, sheep need something to eat, and the amount of sheep that you can make off of some plot of land is always less than the amount of grass because the transfer is not perfectly efficient of the organic matter from the grass into the sheep. You could imagine extrapolating this up further to higher levels of production, like with a 3 and a degree symbol, you'd call this tertiary production. And here's a fierce wolf, an animal that eats sheep, at least in fairy tales. And uh, a- and the thing is, though, when you're talking about the production of animals, it gets difficult to exactly define trophic levels. It's easy to define a primary producer because it's not eating anything. It's just taking raw ingredients from the environment, using an energy source and creating organic matter that way. An herbivore is relatively easy to define because it's something that only eats primary producers. But what if that sheep is eating some bugs along with the grass by accident? Then is it really at the second trophic level, or is that trophic level 2.01 because there are some bugs that it's eating, and maybe the wolf or bear that's eating the sheep, it might also eat some berries or plant material at some point, and so basically, if you're at any trophic level other than trophic level 1, it's hard to perfectly define that trophic level. And the way scientists deal with that is instead of saying secondary production, tertiary production, quaternary production, so on up the food chain, we just call everything at the levels higher than 1 secondary production. And we'd say like secondary production of sheep or secondary production of wolf, and we don't mess around with terms like tertiary or quaternary production normally. I hope that helps you understand what secondary production is. So here's the official definition of secondary production. change in living mass of a heterotrophic population or group of populations, in other words, a trophic level, over time. So heterotrophic means not autotrophic. Autotrophs are primary producers which make their own food, basically like a plant, whereas heterotrophic things are getting their food, their organic matter, from some existing organic matter source rather than making organic matter from scratch. And the way that heterotrophic organisms can increase their biomass, can increase their amount of organic matter, is via growth or reproduction. So uh, an individual organism can grow in size, or it can make more organisms, and both of those can lead to an increase in the living mass of the population or trophic level that you're focused on there. So secondary production is also defined as the heterotrophic equivalent of net primary production. So when we talked about primary production, we talked about how there's gross primary production, which is the total amount of organic matter made by photosynthetic organisms over some period of time. But then you have to account for the fact that there's respiration going on all the time, and a lot of the organic matter is being respired even by the plants. They need to burn some of their own organic matter to keep themselves alive. So the resulting net primary production is some lesser amount than the gross primary production. And when we're talking about heterotrophic production, we only talk about the total amount that's made. We don't usually, we have separate ways of quantifying, like the amount of organic matter consumed. So if you think about this like a sheep, you could measure the total amount of grass that the sheep ate, and then you could measure the uh, increase in mass of the sheep, and the increase in mass of the sheep would be somewhat less than the amount of grass that it ate, quite a bit less actually, because a lot of the grass that it ate would be burned through the sheep's respiration, and or not fully digested and excreted, and both of those things would lead to uh, sort of the mass of the sheep being less than the amount of organic matter that it actually consumed. Okay, so in the sea, moving on from our terrestrial example where we had grass and sheep and wolves, what would be some examples of secondary producers? Well, basically any animal in the ocean would be a secondary producer. So it could be a fish, a squid, a clam, uh, shrimp, copepod. Any animal in the ocean could be considered a secondary producer. So um, to figure out like what we can actually quantify as secondary production in terms of what's going on with a heterotrophic organism, I've included this diagram that helps to explain it. So this is a snail. It could be a sea snail or a land snail, or it could be any heterotrophic organism. All heterotrophic, well, not all heterotrophic organisms, because decomposers don't really ingest food, but any consumer, any heterotrophic consumer ingests food, and you can quantify the amount of food ingested as ingestion. But secondary production is not exactly related to ingestion, because a lot of what's ingested initially ends up being excreted as feces. And um, only a subset of what's ingested is actually assimilated into the organic matter of the organism that ate that food. Of what's assimilated into the body of that organism, much ends up having to be broken down again to power that organism. Even a slow-moving organism like a snail needs to burn energy to be able to crawl and chew and do all the things that a snail does. And so a lot of that organic matter is respired, turned back into carbon dioxide. Um, and the remainder of what's assimilated and not respired is what can accumulate as biomass in that organism and lead to its growth, or can lead to its reproduction. So the making of new snails. And so both the increase in biomass and the reproduction of the organism can be considered to be part of that organism's secondary production. Basically, you've created new snail mass, either in the form of a larger snail or a bunch of baby snails, and that would be your secondary production of snails. Um, and some of those snails that are produced may be eaten, but that's just how it goes. That's still considered part of the secondary production, even though it may not last long before getting eaten by a snail kite or some other organism that eats snails. Okay, so the main way that we measure secondary production is based on biomass. Biomass is kind of an intuitive word because mass, means weight, and bio means alive or living things. So biomass means like the weight of living material. The thing is that we use different ways to quantify the amount of living material depending on whether how accurate we need to be and whether we want to keep the organism alive or not. So the simplest way is wet weight. That's where you just plop the organism onto the scale and weigh it. So you pull a fish out of the water, put it on a scale, and weigh it. That gives you your wet weight, often abbreviated WW. The thing about wet weight is in living organisms, a lot of the biomass is from water, and uh, whether the organisms are dehydrated or sopping wet can lead to like a wide variation in the wet weight that you get. So wet weight tends not to be very accurate, especially for an organism like seaweed that can dry out and lose a lot of its weight really easily and would weigh a lot more if it was fully soggy. So wet weight, not the most accurate kind of weight, but it is the only kind of weight that you can do if you want to keep the organism alive. Dry weight, abbreviated DW, is more accurate. Usually to get dry weight, you have to kill the organism and dry it. Standard temperature for drying organisms to get dry weight is about 60 degrees Celsius. So in my seagrass research, for example, if I want to get the dry weight of an organism like seagrass, I would um, put it in a tin, get the weight of the tin, and then dry the tin and the seagrass in an oven at 60 degrees Celsius for a few days until all the water had evaporated away from the seagrass, and then just what's left is the organic matter minus the water, and that's what you're getting when you get the dry weight. Another step that can give you an even more accurate weight is um, to ash the material. So ashing the material means you actually heat it up to a really high temperature, 450 to 500 degrees Celsius. And that temperature is hot enough that it will actually burn away most or all of the organic matter and leave behind only things like the minerals, from sediment that might have been stuck to the organism, or salt that was in the fluids in the organism, whereas all of the organic matter, all the complex molecules with a carbon framework, are combusted away to carbon dioxide. So then what you do once you have the weight of the ash is subtract the weight of the ash from the dry weight that you got earlier, and that gives you an ash-free dry weight. So that's the ash-free dry weight is like just the weight of the organic matter in that organism minus the weight of the inorganic components, like if it's a seaweed that was muddy and it had a lot of dirt and mud on it, the ash-free dry weight would have sort of eliminated the effect of the sediments that might have been stuck in with the seaweed there. So ash-free dry weight, just a little bit more accurate than pure dry weight. Sometimes we want to be even more specific, and instead of just getting the weight of all of the material in a thing, we want to know the weight of just a specific element or compound in the material. So some scientists will measure carbon weight And sometimes we're interested not only in the biomass of the organic material that's been produced, but we're interested in the energy stored in the biomass. As you might have learned in some of your other classes, different types of organic matter contain different amounts of energy. Like a slab of fat contains more stored energy than a slab of muscle tissue. So that can be important to quantify if we're interested in how energy is passing through the food chain, for example. So sometimes when we're looking at secondary production, we look just at these measures of weight, and other times we might actually look at the amount of energy stored in that material, which we have to measure in other ways. Um, so it's not good enough in terms of production to measure just how much there is. We need to know how much that has changed or is changing over time. So for production, we really want to look at the change in biomass. And this triangular symbol means change in. So we want to look at the change in biomass per unit time. Like, did this fish, how much did this fish grow over a year? Did it put on 10 kilograms of weight over that year? If so, that would be its change in biomass over time. And since we're not interested uh, merely in just like what happened to one individual fish that might have grown over time, but we're really interested in uh, like the entire population of fish, or um, the fish that occupy a set area or volume of water, like a square kilometer, for example. We oftentimes normalize our production measurements to a specific area or volume. Uh, when we were talking about primary production, I mentioned having like a meter squared of lawn, and you could measure the amount of biomass produced by the grass in that lawn over a year, over a unit of area, like a meter squared. Um, and you can do the same kind of thing for secondary production, but instead of being like a meter squared of lawn, it might be like a kilometer squared of ocean, and you could quantify what's the biomass of fish that's produced in that patch of water over a year, and that would be your secondary production of fish uh, for that area. Um, you can convert between interconvert between some of these measures of biomass if you know the conversion factors or the approximate conversion factors. For example, um, 13 grams of wet weight of organic matter roughly equates to about 1 gram of carbon. Um, Although it varies depending on how much of that wet weight material is water and what the carbon to nitrogen ratio is in the organic material. So there's a lot of complications to how we quantify and measure secondary production, and I just want you to be aware of some of those complications and how we deal with them. All right, so to measure secondary production requires some techniques that are a little different than the ones that we use to measure primary production. When I was talking about how we measure primary production, I talked a lot about measuring changes in oxygen in the light and the dark, and using oxygen production and consumption as a proxy for the amount of primary production. Measuring secondary production is a little bit different because we can't sort of equate this consumption of oxygen directly with the amount of secondary production. So think about, how would you measure the increase in heterotrophic biomass of a population over time? Well, there's more than one way to do this, and I'll mention three. So the first method I want to talk about, and the one that I'll talk about most in depth, is the cohort method, where you closely monitor a population over time. Basically, you take a lot of measurements of the organisms to see how big they are at the start of the time that you're interested in, and how big they are at the end of that time, and also how many there are at the start of that time period and how many there are at the end. And you can figure out with a little bit of multiplication what the overall increase in biomass of that population was over time. Another thing that you can do with some organisms is you can label their food source with chemical tracers and figure out how much of the food source that you gave them has been incorporated into their bodies after a certain amount of time. And then you can use that to estimate their rate of secondary production, how fast they're converting their food into biomass of themselves. And that's often done with radioisotope labeling, which I'll talk about briefly in a few slides. Finally, if you maybe don't have the time and resources to do the cohort method or the radioisotope labeling method, you can use some of what we call allometric methods, where you don't directly measure secondary production, but you measure some other things about the number and sizes and types of the organisms that are present in the environment, and then you estimate based on known relationships how much secondary production they're probably doing. So it's not a direct measurement, it's kind of an estimate based on theory. So I'll talk a little bit more about the cohort method here with an example of uh, type of squid that's an important fisheries species in the Pacific and is harvested in particular by Japanese fishing fleets. It's the neon flying squid. These squid actually, to escape predators, they leap out of the water and can soar through the air with webs of skin between their tentacles and on their mantle. So it's sort of a similar strategy to what flying fish use to escape their predators. But of course, fish and squid are not closely related at all, and this evolutionary strategy is independently evolved through the process of convergent evolution in these two very different groups of organisms. Anyways, that was a little aside about the biology of these squid. These squid have kind of an annual life cycle. They grow rapidly and they spawn at a particular time of the year. In late summer, mid to late summer is when the new baby squid start to appear and join the population. And the squid tend not to live much longer than 1 year. And so if you have a species where the production of young is happening sort of all at one particular time, like during the spawning season, then the group of individuals that are produced at that time can be referred to as a cohort. Another time that you might hear the word cohort used is if you're like the new first year student class at the university, they might refer to you as a cohort. And you can sort of study that group of people over time, like all of the students who enrolled in Florida Gulf Coast University in 2020. And then you could monitor like the number that stuck with it through their first year and the second year, the number of students from the would be high originally, and then it might go down as some dropped out or things like that. And the same kind of thing happens in natural cohorts. In nature, you start out with a bunch, and then due to death and well, death mainly, the number in the population goes down over time. So in a cohort, you're starting with a lot of individuals and then you see that taper off over time. So even though the number of individuals is going down from the batch that you started with when the cohort first arose, the size of individuals in that cohort might be increasing. And numbers and size are what's shown on these graphs here. So it's kind of obscured by my little video screen here, but the x-axis on these figures is size of the squid. And so these are big squid at this end of the graph and small squid at this end of the graph. And so you can see that there's kind of like a typical squid size in June. And then in July, it's pretty similar. In August, the squid size has increased a bit because things as they age tend to grow. And in September, you can see that this peak has moved here. The squid that are still alive in September are bigger. But you also start to see this other thing appearing in the diagrams here, and that is a new cohort of squid is arising. So this is sort of the old squid that were born last year that are still hanging on. And then this here now is like the new squid of the year, which are, smaller, obviously, when they're born, and there's some real small ones here. But as they uh, mature, they end up sort of replacing the older squid, which, even though they're quite large now, are diminishing in abundance, which is what's indicated on the y-axis here. Um, so uh, this kind of tracking of a cohort through time is something that's monitored a lot for fishery science, where they need to sort of figure out how many of the species that they're harvesting are out there, what ages and sizes are they surviving, should we how long are they surviving? Should we take more or take less for the harvest? Um, so tracking these cohorts through time is pretty important for setting fisheries harvest limits and things like that. And I'll talk a little bit more on the next slide about how we do this math of figuring out secondary production based on cohort monitoring. Okay, so there's two important terms in calculating secondary production with the cohort method. One is N, which is the number of individuals in the cohort. So uh, if the cohort's the entire population of flying squid, or in this diagram here, it's bluefish. So the entire population of bluefish that were produced in some body of water in a certain season, um, that number would start out high and then it would decrease over time. So you're going from a high N at the beginning, and then a lower N at the end of the period that you're looking at, because some of these things will have died due to predation or other causes of mortality. So N always decreases over time for a cohort. But there's this other important term, W, which means per capita biomass. That means the average weight of an individual in the cohort. So the average weight of an individual in the cohort at the time one is pretty small because the fish were just born then. They're not very big. But later on, although there may not be as many fish as there were at the time uh, that you started, each of those fishes that has survived is bigger than it was before. So the per capita biomass, average per capita biomass has increased over time. So the numbers are decreasing over time for a cohort, but the size of the survivors is increasing over time. And so we use these two basic measurements to come up with a little bit more complicated measurements, delta W and N average. Delta W is the change in per capita biomass over the time interval from T1 to T2. T1 meaning time 1, T2 meaning time 2. So as you pass through this time interval, the surviving fish grew larger, and so you take the weight of that the fish were at the second time period and subtract it from the weight of a typical fish at the beginning of the time period, and that's how you get your delta W. And the other term that you get is calculated a little bit differently. It is the mean number of individuals that were present during that time interval. And so mean is just another word for average. So what you do to figure out the average number of fish that were present in this area during that time interval is you take the number that there were at time one, which you presumably measured with a net survey or some other way of counting fish. And then you take the number of fish that were present at the end of that time interval, and you take the average of those two numbers. So you sum them up and then divide by 2 to figure out what the mean number of individuals was that was present during that interval of time that you're focused on. And then your production during that interval of time, production, remember, being the change in heterotrophic biomass of a population over time, is the average number of individuals that were present during that interval of time times the average change in weight of an individual during that time. So you just multiply those two things together and it gets you your production. Basically how much fish meat was made in that chunk of time. So what we typically do to get more accurate measurements of production is we, instead of just using one time interval, we use a whole bunch of time intervals, and we measure the amount, the population size N, and the typical weight of the individuals W uh, at each of these points in time. And then we can get a production estimate for each of these intervals, and then sum up all of those individual production estimates to figure out the overall production over a longer period of time, uh, like a year. So if you want to be more accurate, you use smaller intervals, and that'll give you a better estimate of the overall secondary production over the longer interval. The thing is, it ends up being a lot of work because, to estimate a population is not easy. You would need to do lots and lots of surveys to accurately estimate the population at all these different time intervals, and um, so sometimes to get a rough estimate, we use larger time intervals. Okay, so now we can talk about one of the other methods for measuring secondary production, and that's radioisotope labeling. So this is one where you create a food source that has some kind of chemical in it that you can trace as that food source is uh, consumed and assimilated by the heterotrophic organisms that you're interested in studying. So you feed that labeled material to the organisms whose production you want to measure, and then you can basically separate the organisms from whatever leftover food there is after a certain amount of time, and see how much of the labeled material that they've incorporated in their bodies. One of the most common types of labeling material used in these studies are what we call radionuclides, like carbon 14, abbreviated with this 14C. And so we can actually easily measure the amount that's been incorporated by measuring the level of radioactivity in the animals or whatever other heterotrophic organisms we're looking at. They could be bacteria or something. In my picture, they look like little octopuses, but more commonly, this technique is used not on multicellular organisms like octopuses, but on simple microbes. Um, anyway, that's the radioisotope labeling method. Another method that's used that's a little easier to apply but has some more uncertainty involved in it is the allometric method. And because there's some uncertainty and some assumptions involved, some people call this voodoo science. Um, and this is a picture of a Haitian voodoo altar. Um, uh, and anyways, that's why I have a picture of a Haitian voodoo altar here. I don't mean to impugn allometry or Haitian voodoo. Um, I just mean that there is a little more uncertainty in measuring secondary production by allometric methods than there is with some of the uh, more direct observational methods like the cohort method or the radioisotope labeling method. But in a lot of situations, the allometric methods are the only way that you can measure secondary production or that you can estimate secondary production. So, knowing that it may not be perfectly accurate, we still do our best, and we have a pretty good idea in some cases of how different factors relate to secondary production. So if we measure those factors, we can estimate secondary production that way. So I ought to explain what allometry is before I start talking about allometric methods. So allometry is the science of relating body size to other things that are affected by body size, like shape, anatomy, physiology, metabolism, and production is kind of like a result of metabolism, digestion, assimilation, and growth. So it's the production is affected by things like the size of the organisms, whether the organisms are warm-blooded or cold-blooded, what the shape of the organisms is, just as other things are affected by size and shape of organisms. This is a picture. I don't know if this is from an old sci-fi horror movie or what. I got this picture off the internet, but basically it shows a very large ant. You don't actually see ants this large on planet Earth because the body structures that work well for an ant that's ant-sized, um, ant-sized, they do not, they would not work well for an organism that was the size of a cow, like this ant is here. It would collapse under its own weight with those twiggy legs. And that's sort of because of the size scaling of things like the surface area to volume ratio, which we talked about in the primary production lecture. So um, because of the way things scale with body size, um, certain things change and it sets limits on life. Like the reason you can't have a Godzilla-sized dinosaur is because it would collapse under its own weight and it wouldn't be able to diffuse the heat that was generated within its body. And for many other reasons, there's sort of like limits on size and body structures. And these sort of constraints on how organisms are built and how they function internally that are related to size also affect their productivity, their secondary productivity. So, giving an example of a really important and well-studied allometric relationship in nature, and that is the relationship of body mass, which is, size, abbreviated capital M, the relationship of body mass to calories burned per hour. So, as you would imagine, a bigger organism burns more calories per hour. Yeah, an elephant eats more than a mouse does. But the rate of scaling is not directly proportional to mass on a one-to-one ratio. So, uh, let's say, an organism that weighs 100 kilograms, which would be like a large human, they don't eat 100 times as much as an organism that weighs 1 kilogram. It's actually somewhat less than that. So the fact that these larger organisms don't have a one-to-one proportionately larger or faster metabolism or bigger metabolism than smaller organisms means that the rate, the factor by which um, respiration is proportional to body mass is 0.75. It's not a one-to-one thing. So basically, that means that if you increase the mass by 100 times, the metabolic rate might only increase by 20 times or so. And we know this because we've measured the size and metabolic rates of many different organisms of many different types, and that's what's indicated by the little black dots that are plotted on this chart. So by making those actual empirical measurements of many, many organisms of different sizes and what their metabolic rates are, we're able to sort of trace or fit regression lines to these relationships and come up with a general um, fairly consistent relationship between the mass of the organism and its metabolic rate. One of the interesting things to see here is that different types of organisms, the type of organism, not just the size, affects this relationship. So for unicellular organisms, they have a relatively lower metabolic rate for a given size than multicellular organisms do. So there's kind of like a shift between the multicellular organism line and the single cell organism line. And also warm-blooded organisms, homeotherms, have a higher metabolic rate at a given body size than um, cold-blooded organisms do. So a giant tortoise might weigh about the same as a panther, but the giant tortoise burns a lot less calories per hour than the panther does because of its cold-blooded lifestyle. So it's not just the body size that comes into play in this allometric relationship, but also the type of metabolism is important to know. Okay, one of the general rules that we got from the previous diagram there is that uh, pound for pound, and I don't like to use non-metric units, but it's a colloquial expression. Pound for pound, smaller organisms have a higher metabolism than larger organisms, and smaller organisms therefore sort of have more rapid production uh, relative to their body size than larger organisms do. And the way we can see that difference is by comparing the production to biomass ratios of small and large organisms. The production to biomass ratio is basically just the amount of production, which is like grams produced over a certain time, divided by the um, starting biomass of that organism. So that would be P divided by B. So let's compare that for two organisms. So at the start of our measurements, we had one small yellow fish that weighed 1 gram and one big blue fish that weighed 10 grams. So over time, the little yellow fish gained 2 grams, and at the end of our measurements, it weighed 3 grams. Over that same time period, the big blue fish gained 5 grams. That's more grams than the little fish gained, and it weighed 15 grams at the end of that time period. Um, so, uh, in absolute terms, the larger organism gained more biomass. But in relative terms, in terms of the production to biomass ratio, the smaller fish made bigger gains. It has a higher production to biomass ratio, 2-to-1 ratio, than this 5-to-10 ratio or 0.5 that the larger organism had. And uh, if you wanted to make an extreme comparison, you could think of something like an anchovy, a small fish which goes from a microscopic egg to a modestly sized fish in just a year, increasing its biomass by a factor of thousands. Whereas something like a whale, it might only gain a couple percent in its weight over the same time period that the small fish was increasing its weight by 1,000-fold. So definitely production to biomass ratio is a big deal in understanding marine life and the productivity of different trophic levels, different levels in the food chain. Okay, so one of the ways that we apply allometric relationships to estimate secondary production is by measuring the size, number, and type of organisms that are present in the community at one instant in time. And so instead of having to make a bunch of measurements over a long period of time and compare them, you just take sort of a snapshot of what's in the community. Basically, you take a scoop of ocean water or a grab of seaweed, and you shake the animals out of it, and you count them up, and after sorting them by size, and you can figure out like how many there are of what different sizes, and with some knowledge of other factors too, like water temperature, which affects productivity because generally secondary production is more rapid at higher water temperatures. You can sort of punch those numbers into some pre-derived equations to estimate the amount of secondary production that was happening in that community. So, like typically small organisms grow and at a certain rate, and we know how that's affected by temperature and by the type of small organism. So if we have those numbers by size class, we can put them all together and estimate the productivity of some small organisms like these ones here. These are amphipods. Amphipods are a common organism in many parts of the ocean. Some of them are planktonic. The ones that I study as a seagrass ecologist are benthic. They live in seagrass beds, and they come in various sizes. The way that you sort of, rather than having to measure each little amphipod with a ruler, to get their sizes, we usually pass them through a series of sieves where the big ones stick in the large sieve and then smaller ones in the smaller sieve sizes, all the way down to the smallest sieve size. So these kind of sequentially filtering organisms through sieves to sort them by size before you count them. These kind of techniques are used by people who study plankton and benthic invertebrates pretty commonly. And one of the reasons that they do this is to calculate with allometric methods the biomass and productivity of the organisms, because you don't just want to know how many there are, you want to know what their productivity is. Why would you want to know what the productivity of, the little shrimp-like organisms are on the seabed? Well, a lot of these little organisms end up being food for things like fishes, including fish that humans might want to catch. So we want to know not just the productivity of the fish themselves, but sort of what the productivity is of the things that could be fish food to know maybe what environments are better habitats for uh, supporting fish life. Okay, lots of different things affect the amount of secondary production. I mentioned temperature was one. Food availability is a huge factor affecting secondary production. You can't have any secondary production unless you have primary production. So if you're looking at the secondary production of zooplankton, the animal plankton, it's going to be really low if you're in an area where there's not much primary production of plant plankton, phytoplankton, right? Um, Another thing that can affect secondary production is predator pressure. So if predators are eating up, like if there's wolves that are eating up all the sheep and the sheep population is really low, then the secondary production of sheep is going to not be very high. Then again, sometimes predation can actually increase secondary production because if it keeps the population low enough that the organisms are able to have plenty of food and grow rapidly, then their secondary production might actually increase. So uh, predation can positively or negatively affect secondary production of the prey species. Something else that affects secondary production is how efficiently organic matter is transferred from one trophic level to the next. So going from one trophic level to another is not a perfectly efficient transfer. We'll talk about that later, but a lot of the organic matter that's consumed ends up just being wasted or respired and not becoming the secondary production of the next level. And the efficiency of that process varies depending on the physiology of the organism eating the prey and on the nutritional quality of the prey, and many other factors can affect trophic transfer efficiency as well. And then there's size and metabolism. So that relates back to the things that we were saying about allometry. Um, Allometry affects trophic transfer efficiency and the rate of productivity. Finally, because we know that some organic matter is lost in every trophic transfer, the um, number of trophic levels that the organic matter must pass through to get, for example, from the phytoplankton all the way up to the apex predators like the sharks. As it goes through all these transfers from one organism to another organism to another organism up the food chain, uh, a lot of the productivity is, or a lot of the biomass is lost. So ultimately, the amount of biomass and productivity of the organisms at the top of the food chain is lower than at the bottom of the food chain, and that's one of the reasons why the most productive fishery in the world is actually the Peruvian anchovies. Um, they are small fish that feed low on the food chain, so they have a high production to biomass ratio, and um, there's just a lot more food available for those anchovies than there would be for some larger predatory fish, uh, which had to maybe eat the anchovies. All right. So when biomass is produced, the fish grow and reproduce. What happens to that biomass? It it doesn't stick around forever and just continue to accumulate, obviously. Otherwise, the oceans would be overflowing with fish, and the planet would just be covered in fish because of this continuous buildup of secondary production. The secondary production of animal life eventually uh, the animals die, they get eaten. Some of that material is transferred into higher trophic levels. If it's an apex predator, it's may not be eaten by anything else, but it'll still eventually die, and then it's decomposed. The organic matter is sort of regenerated back into raw inorganic ingredients by the decomposers. So nothing lasts forever. The biomass that is produced eventually is consumed or decomposed, and the cycle is reset as raw ingredients are regenerated again. But along the way, the organic matter produced can sort of reach successively higher levels in the food chain and end up supporting these really interesting and charismatic organisms that are high in the food chain. So food chains don't just keep going on forever. Um, they do eventually sort of peter out as you get to the apex predators, but there's a lot of interesting life supported along the way until you're eventually, there's not enough energy left to support any higher predators. The reason that trophic transfer, the basic underlying reason that trophic transfer is not 100% efficient is due to one of the laws of thermodynamics, the idea that in every transformation of energy, entropy increases. Entropy is the disorder of the universe, and that generally means that energy is lost as heat. So um, when the sun shines on the water, phytoplankton are able to capture some of that sunlight energy and store it as chemical energy, but they're only about 5% efficient. And a lot of the sunlight just ends up turning into heat, which will eventually radiate away into space and be sort of lost. Um, the Phytoplankton are eaten by zooplankton, and the trophic transfer efficiencies as you go from phytoplankton to zooplankton vary, but a rough number that's often used is maybe 10 or 15% efficiency. So that means that for um, every 100 tons of phytoplankton that's eaten, about 15 tons of zooplankton could be produced. And the trophic transfer is moving on up the food chain through these cold-blooded organisms, like from zooplankton to small fish to salmon, might also be about 15% efficient. And that doesn't mean 15% of the original amount of phytoplankton. That means 15% of the 15% of 15%, which and 15% of that. So you could kind of imagine how you're diminishing as you go up the chain. And you can see that diminishing in the amount of energy remaining in the food chain as uh, as we apply these percent reductions here. So of 100,000 initial units of solar energy, 5,000 of those are captured, but in the photosynthetic process of the phytoplankton. So there's 5,000 units of energy in the phytoplankton. Not all of that is uh, fully transferred into the growth of the zooplankton. So you're left with just 750 units, which is 15% of 5,000 in the zooplankton, and so on up the food chain until when you get to a high level in the food chain, really only a small amount of the original energy is remaining. And that's why organisms high in the food chain are rare. You can go up even further level to the real apex predators in this food chain. This is a marine mammal, a sea lion, which eats salmon. And we'd been using 15% trophic transfer efficiency when we went from phytoplankton to zooplankton, from zooplankton to small fishes, from small fishes to salmon. But now we're using a much lower trophic transfer efficiency to describe the trophic transfer from salmon to sea lions. And you might have already guessed the reason why the trophic transfer efficiency from salmon to sea lions is lower than from anchovies to salmon. And it's because sea lions are warm-blooded animals. So a lot of the food that they eat needs to be burned to maintain their body temperatures and their active lifestyle. So um, that means that marine mammals are even more rare and don't have a high biomass compared to the lower levels of the food chain and the cold-blooded animals. Okay, so because there's this diminishing of available energy and biomass as you go up each level in the food chain, It has a structure that we sometimes refer to as a biomass pyramid. So if you could somehow like drain the ocean and sort the all the organisms that you found by their level in the food chain, and then you made these piles, the pile of phytoplankton would be really large, and the pile of zooplankton would be smaller than that. The pile of small fish would be smaller than the zooplankton. And so there's kind of this irony that the organisms themselves are getting larger as you get to higher levels in the food chain, but the total amount of organic material that is comprised that is contained in those organisms is less than at the base. So um, the total amount of organic material that's stored in the form of uh, plants on Earth is much higher than the total amount of organic material that's stored in the form of animals, particularly more than is stored in the predatory animals. So I just told you that that's the case, that in a typical biomass pyramid, you have a larger amount of plants than you do of animals. The thing is, in ocean environments, sometimes that's not the case. Sometimes you actually have a larger amount of zooplankton in terms of biomass than you do of phytoplankton. And when that happens, they call it an inverted biomass pyramid. And it's kind of paradoxical. How can the biomass of consumers exceed that of the prey? Because we know that you can never have more secondary production than primary production. So how can you have more biomass of the consumers than of the producers? It has to do with the high production to biomass ratio of the small organisms at the bottom of the biomass pyramid. So even though their biomass at any one time might not be very high, they're so productive that they're rapidly turning over that biomass. And so their productivity is higher than the level above. It's just that their biomass at any one point in time is not higher than the amount above. So phytoplankton, because they're really small, and small organisms tend to have a high production to biomass ratio, they have this rapid turnover rate. Basically, phytoplankton might be able to double their population and biomass in a single day, but it's good that they can do that because they might be decimated there and half of their biomass consumed by the zooplankton in a single day. So they're cranking out production, but it's rapidly being consumed. So they don't have like a large accumulation of biomass, even though they have really high productivity. So if you were constructing your pyramid diagram just to represent um, productivity, then it would look like this, true uh, bottom heavy pyramid. But if you were making the pyramid based on the actual biomass present at any one time, it might be one of these kind of oddly shaped or inverted biomass pyramids where one of the levels is at the bottom is lower than the level at the top. All right, a final thing that I want to leave this lecture off with is a difficult problem. This is a problem that I've assigned to my students as part of their uh, practice questions that might be on the test. Uh, and perhaps some will like to try this here. So this problem is going to be sort of a thought exercise about fish secondary productivity. So fish are heterotrophic organisms. They are consumers, and we can measure their productivity. In fact, we do measure their productivity because we like to eat fish and we like to know how much fish biomass the oceans are producing. So fish can be found throughout the ocean, but they're more abundant in some places than others and more productive in some places than others. One of the features of the ocean environment that you can see in this bathymetric diagram here is that waters near shore tend to be a lot shallower than waters offshore. And there's this kind of a fringe of the continents where the waters are shallow that they call the continental shelves of the world. And so if you can kind of divide up the oceans into the waters that are over continental shelves and then the open ocean waters that are not over continental shelves. So continental shelves are only 8% of the sea surface. So obviously 92% of the sea surface is open ocean, right? Everything that's not continental shelf must be open ocean. Um, and even though continental shelves are only 8% of the sea surface, 90% of fisheries landings, that means the 90% of the amount of fish that are caught and brought back to market by commercial fishermen, 90% of marine fisheries landings come from continental shelf waters. And we can sort of assume that that's because the productivity of fish on continental shelf waters is really high. I mean, there might be other factors that could affect it too, like maybe it's harder for the fishers to get out to the middle of the ocean, but it's not unreasonable to assume that the only reason it's not unreasonable to assume that high productivity of these continental shelf waters is one of the reasons why so much of the 90% of the fish landings of the earth come from these continental shelf waters. Okay, um, so here's the problem, and I'll read it and then adjourn the video, and you can also find this problem in your practice questions and your lecture and PowerPoint that I'll make available to you if you're a student. So part one, continental shelves comprise only about 8% of the Earth's total ocean area, yet about 90% of fisheries landings come from continental shelves. So we're making the assumption here that that's simply because continental shelf waters on a per area basis are much more productive than open ocean waters. That could be because continental shelf waters, as we talked about in the primary production lecture, have higher primary productivity due to the nutrients in those waters due to runoff and upwelling. So it's not unreasonable to think that those waters might have higher primary productivity and therefore higher secondary productivity, and that could be the explanation for why we get 90% of our fisheries production from the continental shelf waters. Anyways, what I want you to do is calculate the ratio of secondary productivity per unit area in continental shelf waters to secondary productivity per unit area in the open ocean waters. I want you to figure out basically how many times more productive on a per area basis are the continental shelf waters relative to the open ocean waters. Like, does 1 kilometer, like how many times more fish does 1 kilometer squared of continental shelf water make compared to 1 kilometer squared of open ocean water? All right, and in this problem, it's actually easier if you do parts 2 and 3 before you do part 1. In part 2, I'm giving you a value for the total fish productivity of the ocean on Earth, 2 times 10 to the 8th tons of wet weight per year. So based on that, you have to figure out the total fish productivity of continental shelf waters and the total fish productivity of open ocean waters, remembering that 90% of fish catches come from continental shelf waters, and we're assuming, I guess, that 90% of fish productivity of the ocean is in continental shelf waters, so that should make it easy to solve this part 2. Part 3, I'm giving you this value for the total area of the Earth's oceans and asking what's the productivity per kilometer squared of continental shelf water and productivity per kilometer squared of open ocean water. For this, you're going to have to solve it in 2 steps. The first step will be figuring out what is the area of continental shelf waters and what is the area of open ocean waters of the Earth based on their percentages of this total. So it would be like 8 and 92% of this total. And then you're going to take the values that you got from part 2 for the productivity of each zone and divide it by the area of each zone to get a productivity per kilometer squared for open ocean and for continental shelf waters. And then you're going to take the values from part 3 and divide them by each other to figure out part 1. So I want you to work that all out for yourselves, and then we'll discuss it in class. Thanks.